Guys, welcome back. This time around we're going to take a look at muscle anatomy. Particularly we're going to focus on skeletal muscle anatomy. Here, if you notice, we have a skeletal muscle anchored to a bone. As by definition, skeletal muscles are attached to bones. Typically an indirect attachment, as we see here, is known as a tendon. If it's a rope-like structure, if it's a flat sheet-like structure, it's known as an aponeurosis. But let's look at the parts and pieces, because we need to know the anatomy of a skeletal muscle so that we can start looking at what it is for skeletal muscle physiologically to contract and do its job. I've given you a flow chart at the bottom of the page that goes from deep to superficial, and that's how we're going to describe it from this view. Deep to superficial, notice the smallest functional unit that we have here is known as the muscle cell and or muscle fiber, as we had from the previous video. It is going to be enclosed by a areolar connective tissue wrapping known as the endomysium. A series of these individually wrapped muscle cells are going to be wrapped yet again by into a unit known as a fascicle. Now that fascicle is going to be wrapped or defined by a dense connective tissue, collagen based tissue known as the perimysium. A series of fascicles then, individually wrapped by their perimysium, is yet again going to be enclosed together to make our muscle. And the outer lining that defines the outermost surface of the muscle, it's going to be a very clear, transparent, dense connective tissue, strong structure known as the epimysium. Now notice here, the epimysium extends out, and if we could see on the back side, we would see that this collagen sheath also extends out to give us the strength that we would find associated with that tendon. It extends out past the muscle and to the bone. So let's back up and see it superficial to deep. A muscle is going to be defined on the outermost layer by a perimysium. What a muscle is composed of is individual units known as fascicles. Each fascicle is going to be defined by an outer dense connective collagen based sheath known as the perimysium. And then within the fascicle, we're going to find individual units called muscle cells or muscle fibers, each one of them enclosed by an areolar connective tissue. Now, truth be told, though, most muscles don't work independent of the rest of other muscles in the surrounding area. But if they're having to do a lot of weight-bearing kind of activities, several muscles are frequently tied together so they synchronize their contraction. If we find that group of muscles working together, they're going to be packaged even more. So each muscle is going to be packaged on the outside surface with what we call a deep fascia surface, so that they're all tied together. So if I was going into a cadaver and trying to articulate out one of these large arm muscles, and I was looking for the biceps, the two biceps are actually articulated together and wrapped by that deep fascia. For me to be able to get to the individual muscles, I'd have to cut through the deep fascia and separate the muscles out that way. Now let's go a little deeper then. Let's look at this muscle cell or this muscle fiber. And let's look at what it is to be a muscle cell and what are the parts and pieces of a muscle cell. Here we can see a depiction of a muscle cell. Notice that the muscle cell is defined as our plasma membrane. Now, when we were in cell, we talked about the plasma membrane as being a barrier, separating the inside from the extracellular space. This is a specialized, though, type of plasma membrane. There's only two types of plasma membranes that come with the name lemma. Lemma means it's a plasma membrane that does actually have an electrical event conducted down its surface. The other one being the nerve. So if we were looking at the nervous plasma membrane, neuron's plasma membrane, we would call it the neurolemma. To separate that from the muscle, then we use the prefix sarcolemma. Now inside of this plasma membrane is going to be the cytoplasm. Now cytoplasm is specialized for muscles too, so we call it the sarcoplasm. Now the vast majority of the cytoplasmic space is going to be filled by these little structures here that look like straws, but they're called myofibrils. We'll look a little deeper at what a myofibril is down here in just a moment. Other things that we would find in a sarcoplasm, though, would include mitochondria. A whole lot of mitochondria, because there's going to be a lot of energy used here in the form of ATP. We would love to make that energy aerobically in the presence of those mitochondria. The more aerobically fit I become, one of the things I do is increase the number of mitochondria that I have stored away in those cells for aerobic cellular respiration. Another thing that I would do is increase the amount of glucose I have stored in those cells and the number of myoglobin molecules that I have in those cells. This myoglobin is a specialized organelle in that it is designed to store oxygen within the cell. Now let's back up just a minute. Why not just allow oxygen to diffuse and float freely in the sarcoplasm? The answer to that is oxygen is actually lethal to us if it's stored as oxygen inside the cell. It's a carcinogenic agent. It's an oxidizing agent, if you want to think of it. So we have to sort of bind it in a way in which it's harmless to us as we store it, and that's what the globin molecule does. Globin, again, is a protein that's found in the myo, found in the muscle, that will store for us oxygen molecules. Now, if I take a cross cut of this myofibril, here's what I get. I get a structure that looks a lot like this. Now, the functional unit of a muscle cell, the part in which we will actually physically be able to measure the contraction, percent of contraction of that cell, is something called a sarcomere. A sarcomere goes from one of these little ziggity lines here to the other. So what are these ziggity lines? Now, if we're up here, we see that if we look on the outside surface of a muscle cell, we see that we have these alternating light and dark bands. Well, the dark bands are also called the A bands. That stands for anisotropic, which means that it's hard to see through it, hard for light to get through it. The light bands are going to be known as isotropic, or the I bands. So down here's our A and our I bands now. Here's the I band, and from here to here is the A band. Now let's take a look first and see what's in the A and the I. Well, if you look closely here, you'll see that you got these red, thicker appearance of fibers, and then you have these blue ones. A myofibril is composed of two types of filaments. They're composed of the thick filaments, the red ones here depicted in red, and the thin filaments that are depicted in blue. Notice that the I band, the light band, only has the thin filament in it. Notice that the A band has both the thin and the thick in it. Now, what's in the thick filament? The thick filament is going to be composed of a protein known as myosin, and the thin filament is going to be a protein known as actin. Now, let's take a little closer look at those. Here is that cut out of this little section right in here a little bit. Notice here's our thick filament, here's our thin filament. This thick filament is going to be made up of myosin molecules, thin filament made up of actin. Now down here is a cutout of that thick filament. Notice the thick filament has all these little bead-like structures popping up. What is this? Well, this is about 200 myosin molecules working together. Now notice right here is what a myosin molecule looks like. It is composed of a head group and a tail group. Now the myosin head group is going to be a double helical polypeptide chain that will be found in the core of the thick filament. The head group, on the other hand, is going to be globular proteins, two globular proteins, and the myosin head group globular proteins have an actin binding site, and where they can, a site in which will interact with the actin molecule. It also has a site in which ATP can bind to, an ATP molecule, and within it, it houses an enzyme that can break down ATP so we can use the energy stored in ATP. That's called an ATPase. Now the thin filament over here, on the other hand, is going to be composed of two strands of these blue beads, as you see going through here. Now these blue beads are two strands of F-actin. Now notice each one of these F-actin, it really looks like two strands of blue pearls. Now each one of these pearls is this structure here, a subunit called G-actin. Now notice the G-actins have this indentation in it that represents the site in which the myosin head group will bind. So each Each G-actin is stringed into these long strands, and each strand is known as an F-actin. So the thin filament has two F-actin strands. Notice that the positioning of those head groups, and notice that at rest, this is a view at rest. At rest, the binding site for the myosin head group is blocked. It's blocked by a regulatory protein. This thing that looks like a piece of spaghetti going through here is a protein, a regulatory protein known as tropomyosin. Now what holds it in place and orientates it is a cluster of three proteins that are working in this complex here, these yellow structures. They're known as troponin. Look a little bit closer and you can see that the troponin actually has three structures to it. They are known as TNI, TNT, and TNC. TNI will hold the actin molecule or the troponin molecule onto the actin. TNT will hold the troponin molecule onto the myosin. And then found between them is going to be a TNC component of the complex. And that's going to be where calcium will bind. Because what we're going to see during the process of a contraction, the first thing we got to do is pivot this tropomyosin out of the way from blocking those binding sites. And the way we do that is we will bind calcium to the TNC molecule. And when we do that, it causes it to change a conformational shape in such a way that it loosens and allows the tropomyosin to slide down. I say that up here as well. At rest, the myosin binding sites on the G-actin are being blocked by the tropomyosin. But when calcium is present inside of that muscle, we're going to see that it causes the troponin to change shape in such a way that the tropomyosin can slide down, exposing these binding sites. The last unique feature associated with the myofibrils is going to be this structure here that we see in blue. This structure that we see in blue is called a sarcoplasmic reticulum. Now, yeah, that sounds like a big fancy word. We call it the SR. But you've heard of endoplasmic reticulums and you've heard of a rough and a smooth. Well, this is just a different form of an endoplasmic reticulum that is found specifically and uniquely only on muscle fibers. They're called sarcoplasmic reticula. Sarcoplasmic reticula do have a functional role, and that functional role is to store and release the calcium that regulates the muscle contractions. Now, at the ends, this would be an end of sarcoplasmic reticulums. Notice that they begin and end at these lighter color structures. These lighter color structures are called transverse tubules. Now, these transverse tubules are sort of like a highway that allows you to go from the surface of the sarcolemma deep down into the muscle cell itself, so that the myofibrils that may be found in not associated with the perimeter but deeper in that muscle can also get the same electrical activity that's coming along the sarcolemma. Key structures here are going to be found in this complex right here called a triad, because right here in the sarcoplasmic reticulum, at the end of it, the terminal ends we call the terminal cisternae. So this terminal cisternae plus this terminal cisternae and the T-tubule in between it is going to be the place we call the triad. Now the triad is going to be the place in which the calcium physically is going to be released from when we get to the physiology. So we'll come back to that triad. Other things that we mentioned already is these cells are specialized to have a high concentration of mitochondria, myoglobin, and glucose. Now that we covered that, it's time for you to step back and look at what we've already covered. Review the overview and take a look at the muscle anatomy. Have a good day.